It's really a pleasure to be here and to be in this magnificent auditorium. And if I was sitting there, I'd be tempted to ask questions all the time. Just push that button. And I'm sure many of you will be, and I welcome the questions. But I've brought a lot of slides, and I will skip over some of them depending on how the discussion is going here. So please understand if I'm skipping through slides, it's just so I can get to some of the more salient points. So what I would like to do is give a brief introduction of why we are interested in this area and of the immune system in general, and then get at some details as to how the microbiota might influence certain components of the immune system in various autoimmune diseases, and potentially also in some a fraction of those cases of autism where the environmental influence may be relevant. So we've been hearing a huge amount about the human microbiome during the last few years, especially in the last couple of years. What you all hear is that there are at least 10 times as many bacteria as total cells in our body in the gastrointestinal tract, particularly in the large intestine. The microbiome genome has been called our other genome. Clearly, we have co-evolved with the microbiome, as have many multicellular organisms that have their own particular types of microbiota that inhabit their bodies. And we now know that the microbiota in our bodies, particularly the intestinal microbiota, has a very large contribution to metabolism, of course, in helping digesting foods, in helping harvest energy from foodstuff, but also in production of vitamins, in equilibrating the cells in our body that are in contact, particularly the epithelial barriers. And this is true particularly in the intestinal tract, but also probably true at other surfaces such as the skin as well. In addition, in the last few years, it's become clear that the microbiota, particularly in our gut, has an enormous influence on our immune system, and I'll be discussing that, particularly the microbiota in the intestine. So just to take a step back here, this is a very general slide about hematopoiesis, and we now believe that many cells involved in hematopoiesis are interacting with the microbiota, and just hematopoiesis is the development of cells in our blood, of all the cells in our blood, the different types of cells. And I'll briefly go through some of these. And they arise from hematopoietic stem cells that in the adult are in our bone marrow. These give rise then to multipotential progenitors that can give rise to so-called myeloid cells and lymphoid cells. In this direction are the red blood cells and the platelets, the megakaryocytes that make platelets. But then there are other multipotential progenitors that can give rise to granulocytes and to myeloid cells and to monocytes and dendritic cells. So these give rise to neutrophils, eosinophils, basophils, but also in another branch to progenitors for monocytes and dendritic cells. And I'll come back to this in a little bit because these are very important cells in our immune system in particular. The monocytes give rise to macrophages. The predendritic cells give rise to so-called plasmacytoid DCs and conventional DCs or CDCs, which form a very dense network in the below the epithelial surfaces, including in the gut. So I'll talk a little bit about these. And then there are the lymphoid cells. And lymphoid cells arise from a common lymphoid progenitor, a CLP. And they give rise to the B lymphocytes and T lymphocytes. Am I not sure why this is making a noise, but the B cells and T cells are cells of the adaptive immune system, meaning that that they can generate an enormous repertoire by means of gene rearrangement, and then they are selected by exposure to various antigens, for example, viral antigens or bacterial antigens, so that they can expand and provide memory. And for example, in the Ebola epidemic now, the key is to activate this T cell response to generate B lymphocytes that can make antibodies against Ebola and clear the virus. And then that's why it's possible to transfer serum from those individuals who have recovered to those who are undergoing the acute phase, and the serum can hopefully be protective. And then there are so-called innate lymphoid cells. And some of these have only been discovered or described in the last 2 or 3 years. The natural killer cells we've known about for a long time. They're very important in fighting off cancer cells. And then there are so-called innate lymphoid cells 1, 2, and 3 that I'll tell you more about that can make very similar cytokines or secreted mediators to those made by T lymphocytes. So we think from an evolutionary point of view, these kinds of innate lymphoid cells may have been the earliest types of lymphoid cells that already had particular functions in terms of their capacity for making certain kinds of mediators. And then the T lymphocytes came on later during evolution, and they acquired the ability to have diverse receptors, but really retained many of the same functions as the innate lymphoid cells. And then there are cells very similar to these cells here, the ILCs, that are so-called lymphoid tissue inducer cells. So this is really just an introduction to the vocabulary that I'll be using as I go through here. And if we now just focus on T lymphocytes, the T lymphocytes can generally be divided most easily between helper cells or CD4 T cells. These are the cells that many of you are familiar with because they are the cells that are targeted by the human immunodeficiency virus. So HIV targets the T helper cells. And then there are cytotoxic T cells or the CD8 cells. And most of the cells that we find at the surface, just under the epithelium in the intestine, are CD4 positive T helper cells. We've known for many years that the T helper cells can be subdivided into different categories according to the functions that they have. So for example, there are cells called T helper 1 cells, and these are required to kill a variety of intracellular pathogens, which include viruses. There are many bacteria that can be intracellular bacteria like Listeria, like Salmonella. But these cells are also implicated in a variety of autoimmune diseases. Then there are so-called T helper 2 cells that make different types of cytokines. And these are very important in fighting off parasitic infections, in clearing parasites or helminths, parasitic worms. But the flip side of that is that these cells can also become activated abnormally and contribute to asthma and to allergies. And in the last few years, it's been discovered that there's another cell type that strangely is called Th17. So there's a big jump here. We have a lot of room to fill here with other T helper cells. But it's called Th17 because it was found to make these cytokines that had been discovered earlier, a cytokine called interleukin-17. And a very closely related molecule called interleukin-17F. So these cells are very prevalent at mucosal surfaces, just under the epithelium, throughout the body. And they they're needed to kill extracellular pathogens. So unlike the Th1 cells, which make interferon gamma to kill intracellular pathogens, these cells kill extracellular pathogens, both bacteria and fungi. But they're also critical for repairing tissue that might get damaged at epithelial surfaces. So they're required for barrier protection, for protecting those epithelial barriers. But again, the downside of these cells is that they are critical in autoimmunity. And in mouse, this was initially discovered in animal models. I'll show you one of those models. But it's clear now in human disease that these cells have a critical role, particularly in Crohn's disease, and in rheumatoid arthritis, and also in psoriasis. And in turn, these cells differentiate in the periphery in response to inflammatory signals that come from the innate immune cells that I told you about in the previous slide. And those innate immune cells, depending on what kind of insult they are confronted with, will make different kinds of mediators. So those mediators will lead to the differentiation of different types of cells according to their need to clear those particular damaging agents. The T helper 17 cells differentiate in response to interleukin 6 and TGF beta, but in addition, other interleukins, interleukin 1 and interleukin 23, have important roles. And I highlight interleukin 6 because I'm going to come back to that, because our connection to the Autism project really comes from that particular discovery. And finally, there are cells called regulatory T cells, and there are different types of regulatory T cells. These called iTreg are the ones that are induced in the periphery, and these cells are absolutely essential to prevent autoimmune disease. So basically, once there is an inflammatory process in response, say, to an infection with a virus, the system needs to be shut down, or else there's going to be ongoing damage occurring, and that cannot be sustained. And so the FOXP3 positive regulatory cells come in. FOXP3 is a transcriptional regulator that defines these particular cells. There are individuals, rare individuals with point mutations in FOXP3, which is an X-linked gene, and those individuals usually have fatal autoimmune disease and they usually die as very young children. And what these cells normally do is to restrain the activity of all these other lymphocytes, and they also exist in a balance with the T helper 17 cells. Okay, so I'm going to talk about the transcriptional regulator for T helper 17 cells called ROR gamma T, which is a molecule that controls the expression of the different cytokines, and I'll come back to tell you that in a moment. But I want to put all these different cells that I mentioned at the beginning in the larger context of what is occurring in the mucosa in the intestine, under the epithelium in the intestine. Now, what's really interesting about the intestinal epithelium is that it's a single cell layer that is constantly undergoing replenishment from epithelial stem cells, and it can very easily be damaged. So there needs to be a mechanism to continually repair that epithelial surface, or else it can lead to increased permeability, and it itself can lead to inflammation if bacteria and fungi and food materials get through the epithelial surface. And these different cells that I've been telling you about together coordinate responses to maintain this epithelial surface. But they do so in response to the commensal flora, to the commensal microbiota. So there are these myeloid cells. I told you about dendritic cells, and those are shown in red here. I told you about macrophages that are shown in blue here. So these are very closely related cells that make a series of different cytokines to activate these cells and keep them in a balance. So the key is to keep a balance between effector T lymphocytes, particularly Th17 cells, and the regulatory T cells, the FOXP3 positive cells. So as long as this balance is maintained, everything is fine. But if there is, for example, a decrease in activation of Tregs, that can lead to inflammation. If there's a decrease in signals leading to the differentiation of these cells, there can be susceptibility to infection. Okay? So in addition, there are the innate lymphoid cells I told you about. In particular, I highlight here the innate lymphoid cell type 3 because they also make the same kinds of cytokines as the T helper 17 cells. They make interleukin 17 and another interleukin called interleukin 22. And these cytokines act on the epithelium themselves. Particularly IL-22 has receptors on the epithelium, and that leads to production of these little red dots over there, which are antimicrobial peptides that basically now feed back on the microbiota to keep the microbiota just in the right composition, and also reinforce the junctions that are made between epithelial cells, thus contributing to the fitness of the barrier. And so these cells also require the transcription factor ROR gamma T, as do these structures shown here, which I'll show you in a moment in a real tissue. These are structures that are called cryptopatches that also receive signals from the microbiota, and those signals are needed in order for B lymphocytes to come in, and the B lymphocytes are shown in blue here. When they make follicles, intestinal lymphoid follicles here, these cells now make antibodies, immunoglobulins that can also be secreted to act on the microbiota and again, to contribute to having just the right composition of the microbiota. Okay, so I mentioned the transcription factor, regulatory factor, ROR gamma T. This is how we got into this entire field about 14 or 15 years ago, into studying the role of this molecule, of this transcription factor in lymphoid cells. So this transcription factor is what's known as a nuclear receptor. So nuclear receptors are ligand-activated transcription factors. The ones that are best known are estrogen receptor, or thyroid receptor, or glucocorticoid receptor. So when the ligand is present, it binds to a subunit of this transcription factor, the ligand binding domain, and that leads to a conformational change that allows the recruitment of a coactivator molecule, such as the one shown over here. And this is bound to DNA through a DNA binding domain, and that now regulates the activation of expression of the various genes, where this is bound throughout the genome. So, for example, in the case of ROR gamma T, we think that there's a ligand related to cholesterol that allows this molecule now to recruit coactivator and target the expression of a variety of genes, including interleukin 17 and interleukin 22. So a number of years ago, we were studying this transcription factor, ROR gamma T, which we initially discovered in our work on T lymphocyte development. And we made a mouse in which the green fluorescent protein was used to replace the ROR gamma T gene in a mouse. So in an animal that has one copy of the wild type or normal gene and another copy that's been replaced with GFP, you see normal development of these cryptopatches that I told you about. So you can see this is the epithelium here in green. These are in blue are dendritic cells that surround the lymphoid tissue inducer cells. And the lymphoid tissue inducer cells, the LTI cells, are stained in green with green fluorescent protein. If we make this mutation homozygous, meaning that both copies have GFP, so there is no ROR gamma T being made, these structures never appear. They totally disappear. You can even see a B lymphocyte here that will creep in here when you get signals from the microbiota. And when we do an analysis for cell surface fluorescence using flow cytometry, on this axis we can see the GFP bright cells that are shown over here. And on this axis we stained for the receptor on the surface of T lymphocytes. What you notice is most of the T lymphocytes are negative for GFP. And these are T lymphocytes scattered throughout the so-called lamina propria in the intestine under the epithelium. But what you see is an intermediate GFP population here that are GFP positive. And that's how we were able to show that, in collaboration with a group at DNEX with Dan Kua, who were also working on Th17 cells, that only the GFP positive cells make interleukin 17 within the cell. This is staining inside the cell with antibody to IL-17. And you see the GFP negative cells do not make IL-17. And if the animal lacks ROR gamma T, these cells are completely lacking. And that allowed us then to do a number of studies to show that ROR gamma T is necessary for a variety of autoimmune diseases, and also to show that ROR gamma T is expressed in a series of different cell types and required for them to make their particular cytokines. Not only Th17 cells, but also I told you about the innate lymphoid cells and the lymphoid tissue inducer cells, also so-called gamma delta T cells. So it turns out that gamma delta T cells, it's a different type of T cell. I'm not going to go into that, but these are cells that are very important also at epithelial surfaces, particularly in the skin. And there are therapeutic antibodies for treating psoriasis that I'm sure some of you have heard about that target the interleukin 23 or IL-17 cytokines. And these basically target this pathway here, but it's these cells, along with the Th17 cells, that contribute to psoriasis. And in fact, there's about a 90% remission rate in psoriasis patients who are treated with these antibodies, just indicating the importance of this pathway in general. Okay? So I'm going to skip through this because it gets into a little too much detail, but it's work done by Jun Hu was a postdoctoral fellow in the lab. He developed a screen that I'm not going to go through to identify molecules, small molecules that block ROR gamma T activity, figuring that we may be able to inhibit the differentiation of these different types of cells that I showed in this slide by blocking ROR gamma T activity specifically. And he succeeded in doing this, and I'll skip through this. And the first drug that he discovered that inhibits very nicely and that binds very tightly to ROR gamma T, as shown here by displacement of a ligand that binds to ROR gamma T, is digoxin. Digoxin is one of perhaps the oldest drug known to humans. It's a cardiac glycoside used to treat congestive heart failure. But it turns out that digoxin also targets ROR gamma T, and we even know a crystal structure that was solved by a group in Japan shortly after our description of this. So we know exactly how it binds within the ligand binding pocket of ROR gamma T, basically preventing the recruitment of the coactivator peptide. And using this assay, we were also able to identify other small molecules that work very nicely in blocking IL-17 induction in human cells. So you can see here, this drug that we identified here works very nicely in blocking the induction of interleukin-17 in T helper 17 cells derived from human blood, from human cord blood. And it's very specific for ROR gamma T, because if we overexpress ROR gamma T or a related factor, ROR alpha, in T lymphocytes, you see the very nice induction of interleukin-17, but we completely block this with this particular ROR gamma T inhibitor, but we don't block the ROR alpha induced IL-17 with that particular inhibitor. So we're hoping that this is going to lead to development of therapeutics to treat various inflammatory diseases that are Th17 type diseases. And this is just a proof of principle in the mouse model. This is a mouse model called experimental autoimmune encephalomyelitis. It's a model for multiple sclerosis, and it's debated how good a model it is, but much of the pathologies that it's observed, the demyelination and the remyelination, the repair, appears to be quite similar to what occurs in patients with rheumatoid arthritis. And we can basically score the severity of the disease after we induce the disease here. Typically at about 2 weeks, there is a peak in paralysis that we can measure because there's progressive paralysis in these animals. And we can show here that in animals that receive digoxin in this case, but we've done it with the other better compounds as well, we can now delay and attenuate the disease process. And that's accompanied by many fewer T helper 17 cells in the central nervous system. So we therefore think that what the T helper 17 cells are doing is promoting this kind of disease, for example, in this animal model. And these are cells that we now know are derived during differentiation, particularly in the intestine, from cells that have the potential to either become pro-inflammatory Th17 or anti-inflammatory regulatory T cells. And these two transcription factors that antagonize each other basically can push the cells in one direction or the other, depending on the kinds of microenvironmental cytokine signals that are present. So this is really where the microbiota is acting, where it's most easily observable, in which the microbiota can push cells in one direction or in the other direction. And we can find these cells that have both ROR gamma T and FOXP3 here in human tonsil. So we can stain with antibodies against ROR gamma T. These are T cells. You see the nucleus staining with ROR gamma T. And with FOXP3, these are regulatory T cells. And you see these cells that express both of these transcription factors. And when we look in the mouse intestine, we see something very similar. Now here, by flow cytometry, we look at CD4 T cells, and you can see cells that express FOXP3. Many of them also express ROR gamma T. And we think these are cells that can go in either direction to become either pro-inflammatory or anti-inflammatory T cells. And in fact, we know that there's a great deal of plasticity here. And I don't have time to really go into this today, but what we know now is that there are multiple transcription factors that set up And this is work that we did in collaboration with Richard Bonneau a few years ago. And Rich is here now. Unfortunately, he's not here today. But Aviv Madar and his lab collaborated with Maria Ciofani in our laboratory to do a multi-parameter analysis of transcription factor binding and dynamics of gene expression in Th17 cell differentiation. And we find that there are transcription factors that we call pioneering factors that basically open up chromatin throughout the genome upon activation of the T cell receptor. And then in the presence of polarizing cytokines to make a Th17 cell, that's when ROR gamma T comes in and basically focuses on a few of these sites to activate the Th17 program. And in the process of doing this analysis, we identified other factors, such as the one shown here, which we call a plasticity modulator, meaning that in the absence of this factor, cells can now differentiate towards either regulatory cells or Th1 cells. And we think that there are normal signals in the microenvironment that can actually mimic what we do when we knock down the expression of FOSL2. So this is the point I just want to make here is this is a very plastic system that can be influenced enormously by microenvironmental signals, and many of these microenvironmental signals come from the microbiota. And here's where I'll make the transition to the microbiota, because this is really the key experiment that got us into it. So again, Ivo Ivanov, who discovered the Th17 connection of ROR gamma T and who's now at Columbia University, decided to do an experiment shown here in which he treated mice with antibiotics because he noted that the Th17 cells were most abundant in the intestinal lamina propria. So he thought that maybe they were influenced by the microbiota in the gut. And you can see here that when he used a combination of antibiotics or just vancomycin, there was really a marked reduction in the proportion of T helper 17 cells, cells making IL-17, in the lamina propria. And in fact, animals that were rendered germ-free, that is, in which they were re-derived in a sterile isolator, have no T helper 17 cells in the intestine. So that told us then that the microbiota are needed to induce T helper 17 cells. And then Ivo made a really exciting discovery that set us on our way. What he noted was, depending on where he bought the mice, where he purchased these mice, they either had lots of Th17 cells or very few Th17 cells. And before we even knew what was going on, he did some scanning electron microscopy of the small intestine in these mice. And what he saw is what you see here. The cells that had, the animals that have a lot of Th17 cells have their ileal villi just covered with this spaghetti-like structures here. And those are filamentous bacteria called segmented filamentous bacteria. And they're completely absent in the colony from Jackson Laboratory. And we were able to prove that these are the bacteria that are the key, because a group in Japan at a yogurt company, at Yakult Yogurt Company, for some reason had animals that were colonized only with SFB. So they were germ-free mice that were colonized with these spore-forming bacteria. These are spore formers, so it's relatively easy just to fractionate spores and put them into sterile mice and just grow just those bacteria in those mice. So they sent us some fecal pellets, and we gavaged the animals with these. So you can see here, again, by FACS analysis, germ-free mice have almost no IL-22 and IL-17 produced by their T cells. Okay, so these are lacking T helper 17 cells. And a week after we gavaged them with SFB containing material, we have massive induction of the Th17 cells. So this is really basically a fulfillment of the Koch postulates that it is possible to transmit the phenotype with a particular microbe from one animal to another. So what is the benefit of having this kind of bacteria in the gut? Well, it turns out that if animals are infected with a pathogenic bacterium that resembles enteropathic E. coli, so of course we all know about enteropathogenic E. coli that occasionally you find in spinach and that can lead to really severe gastroenteritis and is occasionally lethal. Citrobacter rodentium is fairly similar, but it's a mouse pathogen, and it adheres to the surface of the colon. And you can see that animals that do not have SFB have quite a bit of bacteria adherent to the colon. Animals that do have SFB are largely protected from Citrobacter, and they're also protected from the loss of weight that comes with Citrobacter infection. But on the other side of the coin here, I told you Th17 cells are important in autoimmune disease. And there's a really lovely model of spontaneous arthritis developed by Christophe Benoit and Diane Mathis at Harvard. And in these mice, if the animals are kept in a conventional mouse colony, they develop polyarticular arthritis that looks very much like human rheumatoid arthritis. But if the animals are treated with antibiotics, or if they move to Jackson Laboratory, or if they're made germ-free, they don't get any disease. So we actually found out about this when Diane and I were both at the same meeting, and I showed the results about the difference between Taconic and Jackson mice. And she said, oh, we moved our mice to Jackson when we moved from France to Harvard, and they no longer were getting sick. So I said, well, we may be able to figure out why they're no longer getting sick. So sure enough, if you give these mice just SFB, now within days they develop arthritis. And this arthritis is dependent on T helper 17 cells. It's dependent on making interleukin 17. So I'm going to skip forward here to give you an idea of how it is that SFB can induce Th17 cells. And we've done a number of studies with different kinds of bacteria that induce different kinds of responses. So I should point out that very shortly after our SFB work, our collaborator from Japan, Kenya Honda, published a lovely paper showing that a different class of bacteria, some subsets of Clostridia, which are also gram-positive spore-forming bacteria, can induce regulatory T cells in the intestine. So, and then we also know that there are other bacteria that stereotypically induce a T helper 1 interferon gamma producing response. And what we are now finding out is that these different bacteria differ in how they interact with the host. And we don't know exactly yet whether there are specific niches or whether they signal to induce different kinds of macrophages or dendritic cells. What we do know is that these cells, once they capture some pieces of the bacteria, migrate through lymphatics to the draining lymph node, in this case a mesenteric lymph node in the gut, and there induce, according to what kind of bacterium instructed this, they induce Th1 or Th17 or Treg cells. And what we find is that SFB-specific T cells are induced here, and these cells then go back to the intestinal lamina propria, where they can serve a protective function. And similarly, Th1 cells can be induced by Listeria to do the very same thing. So the question then is, why is it that some Th17 cells can somehow distribute more widely and cause disease? And indeed, we know now that this does occur. And we are finding that there are specific types of myeloid cells involved here that are involved in the induction of these different kinds of subsets of T lymphocytes. And I just mentioned briefly at the beginning when I was talking about hematopoiesis about the division between dendritic cells and macrophages. But what I'm going to just tell you now is that it's these myeloid types, the monocyte-derived cells, that are important for the induction of SFB-specific T lymphocytes. So the model we have here is that there are these mononuclear phagocytes, which are monocyte-derived macrophages that take up pieces of SFB and migrate to the draining mesenteric lymph node. And within the first 3 days, they induce ROR gamma T, the program for making a Th17 cell. And then after day 4 or so, these cells begin to distribute back in the lamina propria and presumably elsewhere as well. And we find that indeed that is the case. So if we look at SFB-specific T cells, we find, and I'm not going to show you the experiments that we did exactly to prove this, but these are cells that recognize an antigen of SFB. We can stain T lymphocytes that recognize an SFB antigen. And we find that some 90% of them express ROR gamma T when they are in the small intestine. So these are cells that have the T helper 17 program. And these are cells that get induced there and also distribute to the large intestine. Here it's maybe 70 to 90%, 70 to 80% that are Th17 cells. But even if we look now in the spleen, we find these SFB-specific T cells in the spleen that are also ROR gamma T positive T helper 17 cells. So the question that comes up is this a mechanism for autoimmune disease in which these cells that are induced locally in the intestine can distribute broadly? And is it the specificity of these T cells for SFB that somehow crosses onto onto some self-antigen? That's a question that is not yet answered and that many people are working on, but now we have some of the tools to be able to get to this. Okay, so I told you that the Th17 cells are induced in the ileum, and we know that SFB, this segmented filamentous bacterium, primarily is present in the terminal ileum, and this is a log scale here, so you can see massive distribution of these in the terminal ileum. And a few years ago, when we began to work on this, we found several genes that are dependent, whose expression is dependent on having SFB colonization. And one of these is a gene called serum amyloid A. And the serum amyloid A genes are so-called acute phase reactants that are often found in inflammatory conditions. But you can see that they're made only in an SFB-dependent manner in the ileum, as shown here. And this corresponds with there being T helper 17 cells in the ileum that stain both with ROR gamma T, as shown over here on the x-axis, and also with IL-17 on the y-axis. So these are Th17 cells. But here we got a bit of a surprise because we saw a lot of ROR gamma T cells also induced that were present in the duodenum, where there's very little Th17, and also in the colon, where again there's very little IL-17 being made. So what is going on then? Is there something specific there locally? So this is a depiction of what we see then in response to SFB in the ileum. We now see in the mesenteric lymph node the induction of ROR gamma T, as I told you, at about day 3 or so. And then these cells distribute not only to the ileum, but quite broadly. But it's only in the ileum that you get induction of IL-17, as well as other effector functions of Th17 cells that I did not show you. So could these serum amyloid A proteins be involved, since that's where we see these are secreted molecules that are being made there. And these are so-called apolipoproteins that associate with high-density lipoprotein in the plasma. And we know about these mostly from their production in liver during inflammatory diseases. So for example, people with rheumatoid arthritis can have a very high level of SAA. People with lupus, also people who have an acute infection will have a lot of circulating SAA. And it's very typical to see a 1,000-fold increase in the level of these molecules in the plasma. So they're very highly conserved. They have multiple receptors, including innate receptors for so-called toll-like receptor 2 and a scavenger receptor for lipoproteins called CD36. They also appear to be involved in the transport of retinol. So I'm not going to go much into this, but just to show you that if we now look in the mesenteric lymph node for induction of ROR gamma T after the animals, if we are looking specifically at T cells that recognize SFB, you see after SFB colonization, an upregulation of ROR gamma T in the lymph node in mice that have SAA and also in mice that are deficient for both SAA1 and SAA2. So the priming of the Th17 response program in the lymph node occurs, but there is no induction of IL-17 in the small intestine in the animals that are lacking SAA. So that gives us a model shown here, and that is that when these cells redistribute back to the lamina propria throughout the intestine, you need SAA 1 and 2 to be made here, presumably to condition these cells now to become T helper 17 cells. So, now the last part of the puzzle here that we're still trying to work out is how does SFB induce the SAAs? And I'm going to skip over this, but this involves the innate lymphoid cells. And turns out the innate lymphoid cells are also activated by microbiota through a pathway of cytokines to make the interleukin-22 cytokine, which is required for barrier function, and also to make another cytokine called GM-CSF or CSF2 that helps the differentiation of regulatory cells. So the microbiota in this way, through the type 3 ILCs, contributes to the homeostasis in the intestine. So this just to show that there's induction of IL-22 in response to SFB colonization, and you need the innate lymphoid cells in order for this to occur. So the bottom line is that upon SFB colonization, these myeloid cells signal to the innate lymphoid cells, which in turn make IL-22, which then acts on the epithelium and leads to production of SAAs, as well as a number of other genes that are regulated. And then the induction of the Th17 program leads to distribution of cells throughout the gut and probably elsewhere in the body as well. But it's only where you have the SAA present that you now get the induction of IL-17 or the effector function. So a question that comes up then is this something that is also occurring outside of the intestine, where you might have this kind of a additional signal to induce inflammatory functions? And I told you that the Th17 cells have been implicated in many different human diseases, and several of these are here. I told you psoriasis has definitely been validated. IBD is also validated. Mutations in the Th17 pathway are associated with Crohn's disease, for example. Rheumatoid arthritis is a little bit less validated, but I think that there's a lot of evidence for it, and similarly with multiple sclerosis. So many different diseases here that are relevant. So the question then is, does the microbiota also contribute in some ways when the microbiota is disturbed to these kinds of Th17 mediated diseases in human? And we collaborated with a clinical group at NYU, Jose Scher with Steve Abramson, to look at rheumatoid arthritis patients who are coming into the clinic at Bellevue. And I told you already about the arthritis model in mice where Th17 cells are important. And so we wanted to know, is this also true in human disease? And we looked at the microbiota, their fecal microbiota, and we looked at over 100 different patients and control and healthy individuals. And what we found was really striking. That is, in those individuals, those patients who had yet to be treated, new onset rheumatoid arthritis patients, untreated, 75% of them had one type of bacterium, at least 5% of their microbiota, called Prevotella copri. And in the other populations, including chronic arthritis patients, only about 15 to 20% of them had this. And it turns out, this is just some homologies that we did when we did full genome sequences from these individuals. It's really a bacterium that's most closely related to P. copri, Prevotella copri, which is a gram-negative bacterium, very different from the SFB that I told you in the mouse. And the real question is this a causal relationship, or is there just an association due to the disease causing the microbiota to change here? So what we've been doing, and this is work of Hannah Fellner-Peach, a graduate student in the lab, which is still fairly preliminary, but we're very excited by it, because we've been culturing patient-derived Prevotella copri, as well as a reference strain of Prevotella copri, and have colonized mice after we treated these mice with antibiotics, or in some cases with germ-free mice. I think it's with germ-free mice. We colonize the mice with a Prevotella, we confirm the colonization, and then we look for the induction of Th17 cells and Th1 cells and Tregs, et cetera. And we find that only the patient-derived bacterial strain induces T helper 17 cells, as also shown over here. You see ROR gamma T positive cells here compared to the reference strain that does not induce it. So we hope that this is really an index of what might be going on in human disease, in which case it would give us a handle eventually to try to develop some early therapies following diagnosis. So the question still is, are there particular types of T cells that are elicited by these kinds of bacteria that are associated with human autoimmune disease? And can we in some way manipulate the system to try to change the balance, perhaps by increasing the proportion of regulatory T cells, again through various types of bacterial manipulations? And in the last 5 minutes, I want to turn to the work that we've done on autism that's related to this. So I've told you about IL-6 and TGF-beta being involved in the induction of ROR gamma T positive cells that can lead to protective immune response and also to autoimmunity through IL-17 and the other cytokines. And that the bacteria that reside here in the gut have this influence. And I've shown you also that we can use chemical inhibitors to block ROR gamma T and thus prevent those responses in the production of IL-17. But what we also now are working on is the question of whether the Th17 cells can contribute to autism spectrum disorder and the phenotypes in mice. And so this is the proposition that Th17 cells may work at the intersection of gut microbiota, the immune system, and the development of the central nervous system. So inflammation and autism have a long history, which is a bit spotty maybe, but of course we don't really understand the etiology of autism. We know many genetic associations at this point and genetic causes, but the vast majority of autistic disease is not yet explained. And I think it's generally accepted that there are both genetic and environmental factors that contribute. And there is also evidence pointing to a role for immune dysregulation, particularly maternal viral infections early in pregnancy have long been suggested to be involved, and they've been correlated with increased frequency for autism spectrum disease. Now, Paul Patterson, the late Paul Patterson, who very sadly passed away this past summer, has been working for years on a model called maternal immune activation. This is a model in mice primarily, in which the offspring of pregnant mice that were exposed to either viral infection or just to activation of the immune system, for example, through toll-like receptors with the poly-IC polymers, they exhibit behavioral abnormalities that are very similar, perhaps, to what is observed in autism. And the model is a very simple model. The poly-IC is injected typically at day 12 and a half of gestation. And then the offspring throughout their adult life exhibit behavioral defects. And the three core diagnostic features of autism can be modeled in the mice, and that's social interaction deficits using three-chamber types of analyses, repetitive behavior, which we study by looking at marble burying, and also impaired communication, which can be looked at by measuring ultrasonic vocalizations of the pups when they are separated from their moms. I'll just show you a couple of examples of this. But the reason we really got started in this is because Paul Patterson published a paper back in 2007 in which they showed that in order for this disease to be manifested in the offspring, the mother had to have interleukin-6. So if they treated the mother with anti-IL-6, you can see that the social interaction deficit that they observe here, in terms of how much time the animals spend in interacting with another animal rather than an inanimate object, you can see that this is basically ablated with anti-IL-6, and it's also ablated in IL-6 deficient mothers. And I've already told you that IL-6 is a component of the induction of T helper 17 cells, and that's why we began to look at this. Okay, so asking the question, could it be that this particular pathway is involved in this particular disorder? So in order to do this, we wanted to look at ROR gamma T, because ROR gamma T, of course, is required for these cells to develop, and we have genetic tools that we've developed in which we can eliminate ROR gamma T specifically just in T lymphocytes in the mother. And when we do this, and we look at the offspring of mothers that either have the ROR gamma T or do not, this is what we observe in their offspring. And this is a social interaction test in which we measure how much time the mouse spends in a chamber with another mouse or in a chamber with an inanimate target. And you can see with the poly-IC treatment that there is considerably less interaction here in this mouse. But if we now look at pups of heterozygous or knockout mothers for ROR gamma T, we see that the defect is reversed. And we see the same thing for marble-bearing behavior. The animals have this repetitive behavior after this kind of treatment of the mother, and we basically eliminate this behavior if we look in the ROR gamma T knockout offspring. And very similarly for the ultrasonic vocalization, the system that we use, in that system we see an increase in USVs in the pups. And this is something that's been very consistent for us. And you see that now we eliminate this defect when we look here. So although we don't see it in the heterozygote mouse so much. So this basically checks off here all the 3 different types of behavior that we now alleviate in the ROR gamma T, in the pups from the offspring of ROR gamma T deficient mothers. Now, the question is this mediated through IL-17 and IL-17 receptor? We know that interleukin-17A and F made by Th17 cells as signals through a heterodimeric receptor, and we have mice that we obtained that are deficient for interleukin-17 receptor A. And I'm not going to show you the data here, but the data are not quite as clear-cut as with the mother. When we make the mutations in the mother for the social deficit, we actually alleviate that, but we don't see much of an effect on the repetitive behavior. And we also find some effect when we look at the offspring lacking IL-17 receptor. So that really brings into question as to whether this might be a composite effect both on in the mother and in the offspring. And as I close here, I'll tell you what I think we need to do now. But one of the things that we noticed, and also Paul Patterson had some similar data earlier, is this is through work of Gloria Choi, who helped us with this work. She's at MIT. Was that there are defects in the neurons in the mice, in the offspring of mothers that have this kind of treatment. And you can see here the maternal-induced maternal immune activation leads to a much more dysplasia. You see these larger cells in which the cells here in with MIA offspring have much more dispersed sizes and shape throughout the brain. And I'm not really terribly well qualified to tell you much about this at this point, but it really is a very striking result. So we think that we have an objective function here. So how does this relate to the microbiota? Well, there was a paper at the end of last year from Paul Patterson and his collaborator Sarkis Mazmanian at Caltech, in which they use this model, and they show that in the offspring, there is a dysbiosis. That means that the microbiota is altered, and that leads to new metabolites being made at abnormal levels in the offspring, and those metabolites appear to contribute to the behavioral deficits. Not all the behavioral deficits, but some of them. Now, we don't know where this is coming from, and this has not been studied yet. We are studying this. Is there a dysbiosis in the mother that is induced by the MIA protocol, and that dysbiosis is then passed on to the offspring during the nursing period and during birth and during nursing? Or is there a direct effect on the central nervous system in the offspring, or perhaps on the intestinal system in the offspring, perhaps mediated in part through the IL-17 receptor, that contributes then to a later dysbiosis in this offspring? So these are all questions that still need to be resolved. I think what is clear, however, is that this is a fairly robust phenotype that is dependent on this ROR gamma T positive cells in the mother, T cells in the mother. But now we can really try to connect how this relates to the microbiota in the mother and in the offspring. So obviously, this also relates to many studies ongoing about the microbiome, the microbiota in autistic children, and we're all sort of hoping that we'll be able to get some clear answers, perhaps more similar to what I showed you with the rheumatoid arthritis to lead us in the right direction here. So I'm going to stop here, and I've mentioned many of the people who have been involved in this. Wendy Wang and Teruyuki Sano were involved in the T helper 17 work, looking at SFB-specific responses in the gut. Benny Yang, who's now at South Carolina on the faculty, identified the specificity of the Th17 cells. Ivo was the person who really got us started on the road to studying Th17 cells and the microbiota. And Maria Ciofani did very nice work on the transcriptional network. These are some of our collaborators at NYU who have helped us, and also on the work in the arthritis project. Jose Scher led that, and we had help from Eric Paymer's group at Sloan Kettering, and Curtis Huttenhauer and Nicola Segata at the Harvard School of Public Health, who helped us with all the bioinformatics work and the pathway work. And these are folks who gave us MHC tetramers and also various knockout mice. And the folks on the right are all the folks who helped us with the SFB work in the past few years. And I think the Autism project, again, it's Jun Hu, who really has pushed this project. He now is an assistant professor at UMass. And his wife, Gloria Choi, who is an assistant professor at the McGovern Institute, is working on some of this with us now. And Chuck Hofer, who was at NYU and just moved recently to the University of Colorado, has been helping us with the behavioral studies, and Helen Wang and his group. And this is currently funded by SVARI. So thank you very much for your attention. I'm sorry for going a bit over. And I'm glad to take questions and discuss as much as you like.